Hey everyone, my name is Rachel Miner, and today I'm going to be going over a few basic genetic topics. So, to begin, what is a gene? Well, from the ground up, DNA is made up of these special chemical compounds called nucleotides. You might have heard of them either in middle school or high school, but essentially they are adenine, thymine, cytosine, and guanine. There's a fourth one called uracil, but we won't get into that today. Adenine will always bind with thymine, and cytosine will always bind with guanine. This is due to the special way that these chemical compounds are made up, and so they only want to bind to each other. You will therefore never see an adenine bound to a guanine or a cytosine bound to a thymine. It will always be in these A to T and C to G pairs. These nucleotides combine to form strings of information, kind of like the letters of our alphabet can combine to create words and eventually sentences. The specific string of information that you have is called an allele. This is essentially your genetic specific response to a question. So if I asked you, hi, what's your favorite color? Your response could be seen as your allele for that question. This response can either be non-typical or complicating or simple, and I'll get more into that in the next slide. So for that, what is your favorite color example, just went over the fact that if you said your favorite color was red, that would be a very typical response. But what would be another way to convey similar information? Well, a not typical way of conveying the information of your favorite color would be to compare it to something. Say, oh, my favorite color is the color of a Granny Smith apple. Now, I can still understand that response as a response to what is your favorite color. If I wanted to get you something, for instance, that is your favorite color, I would still be able to find something that is that nice light green of a Granny Smith apple, and you would be satisfied. However, there's a third category, the complicating category. If I was trying to get you an object that's your favorite color, and you told me that your favorite color was running, this would be a complicating response. I would perhaps have to rethink my gift idea and instead try and tailor it around your hobby of running instead of your favorite color. This requires extra steps and is therefore complicating. Now, a genetic example could be, what eye color does this person have? The alleles will determine what eye color someone has, the most typical response being brown. This is the most common eye color and therefore is a very typical response. A not typical response could be the eye color of blue. You don't see it nearly as much as brown, but it is definitely a functional eye color that you see all over the place. Finally, we have a complicating eye color, purple. Purple is the eye color seen in people with albinism, as they are not able to take in sunlight like you or I are able to. This makes it so that their eyes are much more sensitive to sunlight, meaning that they have to take extra steps, like having sunglasses. This is similar to the running example because while in that example you have to take extra steps to ensure that you're accommodating the person, here you also have to take those extra steps to accommodate that eye color. So how do genes work? Well, there's this central dogma of molecular biology that all my professors love to talk about. Essentially, it's that DNA goes to RNA, which goes to proteins. So what does that mean exactly? Well, the best way that I've thought to describe this is that DNA is kind of like an executive. It has everything that it wants to be done in its head, and it just kind of is trying to get those orders out. RNA, meanwhile, is kind of like the secretary and the messengers. As the secretary, it writes down all of the messages dictated by DNA, but perhaps the DNA says or and, or any other sorts of filler words, or wants something stricken from the record. Well, it's RNA's job to do that. RNA can cut out pieces that are irrelevant or unnecessary, and also help enhance things that are relevant or are necessary. It then has certain messengers that will convey this message from the DNA to the workers, which are, in this case, the proteins. The proteins are responsible for building up the various things in the body, and therefore are very much like the workers of our body. They are very much more than just what you get when you eat beans or a steak, but are instead the structures that are created by reading DNA, and they as I said, perform all of the tasks in the body. So how do mutations arise at the genetic level? Well, here I have an example sentence made up of three-letter words. You might notice that there's nonsense at the beginning and nonsense at the end. This is intentional, as much DNA is made up in the same way. You also have an SRT and an STP. That first SRT is going to resemble our stop codon. The second STP is going to represent our stop codon. So mutations at the genetic level can occur in a variety of ways. The least problematic is a base substitution. A base substitution would be like taking our sentence, "the cat ran out the hut," and replacing the C in cat with the B that makes this word now bat. Clearly, the sentences "the cat ran out the hut" and "the bat ran out the hut" are different and mean different things. However, you can still understand the gist of the sentence for both of them. Moving on, we have deletions and insertions. These are some of the more problematic kinds of mutations that can arise. In a deletion, we've deleted the T from our cat in our previous sentence. This leaves us with the "ka ran out the hut." However, DNA does not just stop here, because it actually reads in three-letter groups. And so instead of having a two-letter group, where cat was, and maintaining everything else, it instead shifts everything to maintain that groups of three reading frame. So now we have the car NOUT, and you see how it has completely gone and messed up going forward. Insertions are very similar. If we were to insert an S into the word out, you can see that the entire rest of the sentence gets mixed up. An important thing to note is even the stop symbol gets messed up. It gets incorporated into the other words and therefore is not read as stop. This means that our sentence essentially has no period and therefore has no end, and instead runs into the nonsense that is after the sentence. It is important to also note that mutations are increased by mutagens, and so things like, smoking, alcohol, sunlight even, can create more and more of these mutations and create more and more of these issues. Briefly, I'd like to touch on epigenetics. Epigenetics is essentially outside factors that allow genes to be promoted or suppressed. These are factors that aren't in that code of the DNA itself, but are slightly outside of it. You can kind of think of these as taking that transcript of that DNA creates and either crumpling it up into a ball where it can no longer be read, or opening up a ball that has already been crumpled up so that it's easier to read. Epigenetics can often be reversed, but not always, and are just another way for the body to make sure that DNA is either able to be read or not read when it is necessary. Finally, I'd like to go over the basics of meiosis. Meiosis is the way that you or I was created, and is just basically the simple way that genes are passed on from our parents to ourselves. In meiosis, you have these chromosomes, which are essentially where DNA is stored, and they'll pair up with the similar chromosome from your two parents. They match up and exchange information, kind of like exchanging their answers for a question, sometimes changing each other's minds, sometimes not. And in so doing, you can see that you kind of get a mixing of colors as these various genes have been swapped between the chromosomes that have been paired up. Finally, the chromosomes yet again separate and go into the cells that will eventually become ourselves. Now, this can also get messed up in something called non-disjunction. In non-disjunction, what happens is instead of splitting up like they're supposed to once they've exchanged information, the two chromosomes stick together. They fail to disjoin, and therefore travel together into the new cell. This results in one cell having 3 copies of these chromosomes, whereas it should only have 2. And meanwhile, another cell will be left with only 1. This is very problematic and can lead to various disorders and syndromes, such as Down syndrome. That's it for the basics of genetics. Thank you all so much for watching.